Welcome to the 17th Student Council Symposium of the ISCB Student Council. My name is Cynthia Elizabeth Castillo-Silva, and I'm here to talk to you about the spatial variability of SARS-CoV-2 Spike protein and its effect on COVID-19 disease outbreaks. In late December 2019, a different strain of coronavirus, which caused severe acute respiratory syndrome known as SARS, emerged in Wuhan, China. COVID-19 disease caused by SARS-CoV-2 quickly was spread to other countries, and the World Health Organization declared the COVID-19 outbreak as a global pandemic on March 11th. Transmission occurs via inhalation of the respiratory droplets exhaled from an infected person. SARS-CoV-2 infects type 2 pneumocytes localized in the pulmonary alveoli. In early stages, viral spike glycoprotein recognizes the angiotensin-converting enzyme 2, also known as ACE2 receptor. As the virion binds with the ACE2 protein, another membrane protein called TMPRSS2 cleaves the spike protein at multiple sites. These mechanisms allow entry of the virion, releasing its RNA strand, which is translated into several proteins. However, the mutation rate from RNA viruses is drastically high and provides a competitive advantage for viral adaptation. Clade systems allows us to group these mutations by shared characteristics, but normally it takes time to classify a new mutation, and sometimes there are several clade systems for the same species. On the screen, we can see the most known clade systems for SARS-CoV-2 taken from GSAID's database, where we can see distributions per region and per clade for all their samples. In this work, we will not use the clade system, at least not this one. We will instead perform a random forest algorithm to classify all mutations into characteristic groups. The data we used was from the National Center for Biotechnology Information, also known as NCBI. More than 39,000 complete spike protein sequences until February of this year were retrieved for its database. It is important to note that the distribution of the samples was not equal. Almost 90% Almost 90% of the data was taken from the US and Australia. Once we get all the samples, we proceed to align these samples. After this, a time-to matrix was used to define a metric to score amino acid mutations in these proteins in order to determine their divergence in phylogenetic trees. Then, we divided our samples per region to perform a topological data analysis to find out how different these regions were from each other. From heat map, we show the detected cross-correlations between pairs of datasets. Thus, California, Washington, and Australia belong to the same cluster. This means these regions have sequences with similar mutations. This group has a quite different mutation pattern from the rest of the groups. The east coast of the USA cluster is close to Europe, Western Asia, and Eastern Asia groups. Meanwhile, Central, Southwest, North, and Midwest of the US and South America belong to the same cluster. Although DDA is a robust method for n-dimensional analysis, machine learning analysis is better at discriminating variables and finding patterns. Characteristic mutation clusters of the IS protein sequences were classified using a random forest algorithm and diffusion maps. This allowed us to identify the most important mutations clusters related to the outbreak dates in different geographic regions, as we can see in the figures on the screen. It is important to keep track of the mutations that occur on the RBD domain of the S protein because of a natural adaptation with the host. We found 4 hypervariable motifs in the N-terminal domain, and one more after the RBD sequence. More amino acid variations occur in the regions of high content of NAG, as seen in the picture of the right in color cyan. Several studies suggest that new outbreaks are caused by the appearance of increasingly infectious variants like the Delta variant, for example, of the clade system from GSAID. Topological data analysis allowed us to identify close relationships between mutation patterns, suggesting routes of the spread of the pandemic. The random forest algorithm was applied to detect significant missense mutations, such as S4077N, D614G, L5F, and others. Some of these mutations have been associated with increased infectivity and virulence. And finally, we identified the regions of high variability along the spike protein sequence, particularly localized in the NTD region that has high contents of NAG. This is relevant to assume that the NTD region is involved in evolutionary adaptation. Studying the variability of the spike protein will require significant efforts to develop wider spectrum anti-SARS-CoV-2 strategies. It is necessary to keep ongoing mutational sequencing and monitoring studies, and to avoid aiming efforts to the high variability regions that contain the COVID-19 disease. I would also like to acknowledge scientific and technological research support program, also known as PAICIT, of the UANL for supporting this work, and Cabana for awarding a fellowship to attend this symposium to our team. And finally, I'm honored to introduce you to our research team. For further information regarding this work, please contact Dr. Francisco Hernandez Cabrera to the email shown in the screen. Thank you.
Okay, thank you very much for your presentation. Please send your questions in the QA section, please. Okay. I have a question. So, do you think that in the NTD zone that you described, a mutation could cause a more virulent variant, maybe? What do you think about that?
The NTD region, I don't know exactly, because I'm more focused on the mathematical side of this investigation. So I will say that I'm not sure.
Okay.